Good afternoon, everyone, and thank you, Paul, for the introduction, and Selecta for the kind invitation and all the collaborative efforts over the past years to move RNA from basic biology to therapeutic opportunity in cancer. So the overarching goal of my lab is to fill what we call the RNA gap in cancer. And while cancer therapy has historically focused on DNA or proteins that can be targeted by using chemotherapy or precision and cellular therapy, RNA remains, and particularly non-protein coding RNA, remains largely untapped. And we believe that filling this gap may open to novel therapeutic opportunity in cancer. In particular, our focus is on long non-coding RNA or lncRNA, and these are RNA molecules without or with a very limited protein coding potential that by convention are longer than 200 or 500 nucleotides. The key question that we try to address in the lab are which lncRNA drive cancer, what are their mechanisms, and how this can be targeted for therapy. And in doing so, our main disease model is multiple myeloma, which is a very challenging malignancy of plasma cells. So for today, I will focus on the first question, and I will show what we have done and are doing to discover which lncRNA drive cancer. First of all, why long non-coding RNA matter at all in cancer? As you can see on your left, lncRNA genes outnumber protein coding genes with up to 100,000 lncRNA genes that have been identified so far. And these are not just transcriptional noise. Many functional genomic studies, like the one that I'm showing on the right by Montero et al. Using CRISPR-Cas13, but also our own work with CRISPR-Cas9 and CRISPR interference, have shown that many long non-coding RNA are essential in cancer, but with an important caveat. This body of work only focus or treats long non-coding RNA are single transcriptor genes, and this is the limitation that we need to overcome. Let me explain why. Long non-coding RNA, like messenger RNA, are extensively spliced, producing multiple isoforms, and this isoform, as is intuitive, will have different sequence, will fold in different structure. They may also play different and sometimes divergent function, and they need precision strategies for their targeting. And this is not just theory, but we touched with our hands how isoform diversity matters by working on this non-coding RNA gene called MIR70HG. This gene was very well known to produce a transcript that can provide the precursor for a cluster of microRNA called MIR1792, which drives different cancer types. More recently, what we found is that it can also produce an alternative transcript that we call lncRNA1792, and how this long transcript and the microRNA differ. While microRNA go to the cytosol and regulate gene expression, it is a very well-known mechanism, the lncRNA1792 stay in the nucleus, and in the nucleus can bind different, on the chromatin at different gene loci, providing the scaffold to assemble ribonucleoprotein complexes that then drive the expression of essential pathways. So isoform resolution is key for getting functional and therapeutic insights on long non-coding RNA, and without isoform resolution, we are like this guy. We can identify as many essential lncRNA we want, but we will be unable to proceed and understand what is their mechanism of action and develop precision strategies for their targeting. So today I'm very glad to present our latest work. We developed a novel platform that we call IsoScan, which allows to map isoform selective lncRNA dependencies in cancer. IsoScan combines the unique ability of CRISPR-Cas13 to target lncRNA transcripts with an innovative design of guide RNA, for which I want to thank again Selecta. In this case, guide RNA are designed to bind unique exon-exon junction or unique non-coding exon. And in this way, the platform allows the lncRNA isoform knockdown in high throughput manner targeting lncRNA from any genomic origin and acting in any subcellular compartment. Let me give you a few more details on how we use our platform. IsoScan was applied to a large panel of cancer cell lines, primarily multiple myeloma, in the context of dropout viability screens. For those who are not familiar with this screen, in the dropout viability screen, we measure the guide RNA representation over the course of an experiment, and the interpretation is that those guide RNA that are depleted at the end of the experiment are targeting essential targets, in this case, lncRNA isoforms. And our library was applied to more than 5,000 lncRNA isoforms that correspond to the entire myeloma lncRNA transcriptome as measured in more than 300 myeloma patients with 9 guide RNA per isoform. So let's start with the results. Our primary screen in 5 myeloma cell lines identified up to 600 isoforms that were essential for the myeloma cell growth. Half of this isoform, highlighted in this plot in orange, were essential only in 1 myeloma cell line. The other half were essential in 2 or more myeloma cell lines. In this plot, you can see those essential in 2 cell lines in purple, 3 in blue, 4 in green, and 101 were essential in all myeloma cell lines that we tested and are highlighted in red. And this included important positive controls like MALAT1 or MIR70HG, which are known drivers of myeloma cell growth. To validate these findings, we generated a secondary library called IsoScan L2, targeting those isoforms that were essential in at least 2 myeloma cell lines. And through this secondary library, we demonstrated the reproducibility of our data in vitro, as you can see on the top right, ranging from 80% to 90%, but also in a clinically relevant animal model. So let me explain you better what we mean with isoform resolution, giving an overview of our data. As I mentioned, we identified 598 tumor essential isoforms. These isoforms derived from 479 lncRNA genes. This means that some genes contribute to with more than 1 essential isoforms. However, it's also important to note that this gene can produce more than 1,000 isoforms expressed in the myeloma transcriptome and more than 10,000 isoforms that are annotated across the genome. So without isoform resolution, we would be navigating in the dark, not knowing which of these thousands isoform is actually driving the cancer dependency. This is well illustrated if we focus on a subgroup of lncRNA called small nucleolithium host gene, SNAG. These are named this way because they host small nucleolithium RNA in their introns, but they can also play independent function as long non-coding RNA. As you can see in the heat map, we identified seven relevant SNAG genes that could produce more than 100 annotated isoforms, of which 32 are expressed in myeloma, but only 9 are essential for the cancer cell growth, with each of these genes contributing to at least 1 isoform, but in some cases with more than 1, like you can see for the link point. Again, if we go deeper with one example, let's focus on SNAG6, which is one of these essential lncRNA. This long non-coding RNA had 3 isoforms that were targeted in our screen. Only the isoform 003 was providing significant dependency. As you can see on the right, dedicated experiments demonstrated that IsoScan guide RNA targeting the isoform 0003 were selectively downmodulating this isoform, while the other 2, the number 5 and the number 6, were upregulated. We believe that this upregulation could be a compensatory mechanism or could point out to an inter-isoform regulation. Also, if you see on the far right, we found that the SNORT87, which is hosted in the sequence of this isoform was not downmodulated, again demonstrating that our approach is really transcript selective. On the contrary, the guide RNA that targeted the isoform 006, which is not essential, were proved not to affect the viability of cancer cells and to selectively targeted downmodulating these non-essential isoforms. Overall indicating that our approach is really selective in isolating isoform level dependencies. So now we have shown that we identified hundreds of isoforms that are essential for cancer cell growth. Our approach is selective, but how strong is the dependency on this long non-coding RNA? As you can see from these slides, we identified more than 200 lncRNA isoforms that had a dependency score that was comparable in magnitudes to the positive controls that were included in the screen, like known oncogenic lncRNA like MALAT1, NIT1, MYD70HG, but also comparable to IRF4, which is a protein coding gene, and it is the leading dependency among the protein coding genes in multiple myeloma, according to all the functional genomic studies so far. So here the take-home message is very simple. Long non-coding RNA are as essential as protein coding, and the dependency is driven by their specific isoforms. To broaden our observation, next we applied our secondary library to a larger panel of cancer cell lines that included solid cancer cell lines from colorectal, lung, and breast cancer, and also to 3 lymphoma cell lines. As you can see in these slides, to our surprise, the majority of these dependencies were conserved across the tumor types, with 40% of them being essential in all or most the cancer cell lines that we tested. On the other hand, only a minority, highlighted in blue, proved to be selective for multiple myeloma, and this is important because it is in contrast of what we were thinking about lncRNA that are strictly tissue-specific. This observation can be partly explained by looking at their expression level. If you see in this slide, in blue we have myeloma selective isoform, and you can see that these are significantly more expressed in myeloma versus non-myeloma cell lines, while the pan-cancer isoform show a very similar expression level. What is also important if you look at the plots on the pan-cancer isoforms is that their expression can be really heterogeneous. We have isoforms with really high expression and isoforms with really low expression. This means that while expression could be important, it cannot explain alone the dependency on this long non-coding RNA. So next, we try to get some biological insights on this essential long non-coding RNA, looking at the localization and the site of dependency. To localize where these lncRNA are expressed, we use a compartmental RNA sequencing, which is a technique which separates chromatin-bound, nuclear-soluble, and cytoplasmic RNA before the sequencing, and then allow to map the relative expression of each long non-coding RNA in these 3 compartments. As you can see from this plot, majority of these lncRNA isoforms were more enriched in the nuclear compartments, either chromatin-bound or nuclear-soluble, but a small subset was also particularly expressed in the cytosol. To understand where they play the function, so where they are essential, we repeated our screen using a Cas13 that localized to the cytosol or a Cas13 that remains in the nucleus using a nuclear export or nuclear localization signal. As you can see in the plot, and consistent with the localization study, majority of these isoforms remain essential only when targeted in the nucleus, and only a small subgroup was proved to be essential when targeted in the cytosol. So now I will focus on one example to show how localization is important to understand the mechanism of action, and I will focus on the on this isoform of SNAG6 because it is very peculiar, was one of the few lncRNA enriched in the cytosol and a leading dependency when targeted with the cytosolic Cas13. So using super resolution microscopy, what we found is that this isoform is not just diffusely expressed in the cytosol, but specifically enriches in the endoplasmic reticulum. And consistent with this localization, what we found is that this long non-coding RNA directly interacts in living cells with many proteins that reside in the endoplasmic reticulum, including the heat shock proteins that are also very relevant to maintain ER homeostasis and protein homeostasis. And again, consistently with the function, with the localization, we found that knocking down this isoform, but not the alternative isoform 005, triggers the markers of ER stress and unfolded protein response. Overall, these data are important because they are linking the localization of a long non-coding RNA isoform to its mechanism of action. So far, I've shown functional data obtained in preclinical models. Our next question was, are these long non-coding RNA isoforms also relevant for the pathogenesis of myeloma, and can they affect the clinical behavior of myeloma patients? To address this question, we analyzed RNA sequence data obtained in a cohort of 316 myeloma patients that were clinically annotated and homogeneously treated. As you can see from the circus plot, 85 of these isoforms were more expressed in myeloma versus normal plasma cells. 44 were specifically upregulated in patients with high-risk genomic features, including those with the amplification of chromosome 1q or deletion of the 17p. And for 92 isoform, we found that higher expression was correlating with a shorter overall survival. In short, this data mean that these long non-coding RNA are not just essential in preclinical models, but they are involved in the pathogenesis of myeloma, and they can have a real clinical significance. So finally, we have integrated all this data in a friendly user interface that we call the LongDep portal. This is a portal that is meant to allow any researcher around the world to freely browse our data and start the research on long non-coding RNA. I will explain very shortly. There are two main items. Through the clinical items, a researcher will be able to navigate all our expression data obtained in myeloma patients, and will be able to know which long non-coding RNA isoform is differentially expressed in myeloma versus normal plasma cells, if it is differentially expressed in any myeloma subgroups, if it has any effects on the clinical outcome of patients. Through the functional item, we'll be able to navigate all our functional data that I have explained so far. And to conclude, the IsoScan platform now enables us to define tumor-essential lncRNA at the isoform level, locate where this isoform exerts their function, and gain insights into their biological roles. If we go back to the frustrated scientist, now he or she has a tool to identify which isoform is driving the phenotype, understand the mechanism of action, and hopefully develop the precision strategy for the targeting. Before concluding, I just want to give a little insight on what is next, what we are doing now. Again, with thanks to the collaboration with Selecta, we couple our IsoScan platform with single-cell RNA sequencing in an approach that we call isoperturb sequencing. Through this approach, we have been able to dissect the transcriptional pathway that are modulated after knocking down 155 isoforms that are essential in multiple myeloma. I hope I will have other opportunity to discuss this data. And thank you for the attention, and I'm open for the question. Before that, please let me acknowledge all the people that have been collaborating, in particular Dr. Maisano and Akhtas Samour, that have led with me the development of IsoScan. My mentor for life, Nikhil Mushi, at the Dana-Farber Cancer Institute, all the society that have been funding our research, including AIRC, Fondazione Piemontese per la Ricerca sul Cancro, Leukemia and Lymphoma Society, and the Paola and Roger Rainey Foundation, and of course, again, all the team at Selecta for their support. And thank you.